All right, ready to dive deep today
Always ready.
into the world of drug discovery.
Sounds good.
We're tackling hit-to-lead and lead optimization. I know it sounds a bit, well, technical, but believe me, it's fascinating stuff.
Yeah, it really is.
It's kind of like, taking this rough gemstone
Right.
and carefully cutting it, polishing it until you have this dazzling, valuable jewel.
I like that analogy.
We're talking about taking a molecule that shows a glimmer of potential against disease.
That's our hit,
Exactly.
our starting point.
And transforming it into a powerful contender.
That's the lead.
Our lead, you've given me a ton of info on this.
Oh, yeah.
Our mission is to sift through it all and
Great.
extract the most important insights and help you understand what really matters.
Sounds like a plan.
So it's not just about finding a molecule that works against a disease, right?
Right. It's got to function as a medicine
Ah.
inside the human body.
Got it. So what's this balancing act all about? What are researchers trying to optimize when refining these molecules?
So they're really focusing on three main things:
Okay.
potency, safety, and what we call drug-like properties.
Potency, safety, drug-like properties. Let's break those down one by one,
Sure.
starting with potency.
Potency is all about how well the molecule interacts with its target. Think of it as the drug's strength,
Okay, the drug strength.
how effectively it can do its job.
Makes sense.
And safety. Safety, of course, is about minimizing any potential side effects. We don't want that cure to be worse than the disease.
Yeah, definitely not.
So that's a major consideration throughout the whole process.
Right. And then there's drug-like properties. That one's a little less obvious. What exactly does that mean?
So drug-like properties are all about making sure the molecule can actually be absorbed by the body, distributed to the right places, metabolized effectively, and then eventually eliminated safely.
So it's like making sure the drug can navigate the body and get to where it needs to go.
It's like making sure the drug can navigate the intricate highway system of the human body and get to its destination without causing any traffic jams or accidents along the way.
I love that analogy. So it's not just about hitting the target, it's about making sure the drug can travel there safely and efficiently.
Exactly. And to achieve this balance, researchers use a variety of techniques. One of the most fundamental concepts here is SAR, which stands for structure-activity relationships.
SAR. Okay. So how does tinkering with the structure of a molecule actually change how it behaves?
It's a lot like, you're an architect designing a building.
Okay.
Even a small change to the blueprints, the
Right.
position of a wall, the size of a window, can have a huge impact on the final structure.
Right.
It's the same with molecules,
Uh-huh.
even tiny adjustments. Adding or removing a single atom can drastically alter how the molecule interacts with its target, how it's absorbed, metabolized, and even its potential side effects.
So chemists are like molecular architects.
Exactly. They're making these tiny adjustments and observing how they change the molecule's behavior.
That sounds incredibly intricate.
It really is. It's a delicate dance, and sometimes even a seemingly minor tweak can make a huge difference.
I can imagine. So we're talking about a lot of trial and error here.
To some extent, yes, but it's not completely random. There's a lot of strategy involved. One study we looked at focused on metabolic stability during lead optimization. They found that a lot of promising candidates were being dropped because they were metabolized too quickly in the body, meaning they wouldn't stick around long enough to be effective.
Oh, I see. So a drug might be potent in a test tube, but if it gets chewed up by the body's enzymes before it can reach its target, it's essentially useless.
Exactly. And what's interesting is that this screening process often only focused on the original parent compound.
So they weren't looking at what happened to the molecule after it was broken down by the body.
Right. They were missing a crucial part of the picture because sometimes those metabolites, the molecules it breaks down into, can actually be pharmacologically active themselves. They might even have better drug-like properties than the original compound.
So a molecule that seemed like a dud because it was rapidly metabolized could actually be a hidden gem. That's a fantastic example of how important it is to consider the entire journey of a drug within the body, not just its initial form.
Absolutely. And this complexity is amplified when we consider the different approaches to lead optimization. On one hand, you have predictive modeling, which uses computer simulations to try and predict which molecules will be most effective.
So it's like a virtual laboratory trying to speed up the discovery process.
Exactly. It's a way to explore many different possibilities without having to synthesize and test each one in the real world. But then you also have empirical optimization, which involves actually synthesizing and testing those molecules in the lab, good old-fashioned trial and error.
So it's like weighing the pros and cons of a virtual scouting mission versus actually venturing out into the field.
That's a great analogy. And the choice of approach really depends on the specific target, the available resources, and the desired timeline.
So there's no one-size-fits-all approach.
Not really. Sometimes it's a combination of both. You might start with predictive modeling to narrow down the possibilities, and then use empirical optimization to fine-tune the most promising candidates.
So you use the virtual world to guide your exploration, and then the real world to confirm and refine your findings.
Exactly. It's a powerful combination.
Speaking of real-world applications, you mentioned COVID-19 antivirals earlier. I'd love to dive into a specific case study where lead optimization played a crucial role in developing a treatment, or maybe even where it presented significant challenges. Do you have any examples that come to mind?
Absolutely. The development of SARS-CoV-2 antivirals is a perfect example.
Okay.
It was a high-stakes race against time, and lead optimization played a critical role in both the successes and the setbacks.
Okay, so let's dive into that. What were some of the key hurdles researchers faced when trying to optimize antiviral leads for COVID-19?
One of the biggest challenges, as we touched on earlier, was balancing potency against safety. You want a drug that can effectively stop the virus from replicating, but you also don't want it to cause harmful side effects. And finding that sweet spot can be incredibly difficult, especially when you're dealing with a novel virus.
Right, it's a delicate tightrope walk.
Yeah.
Were there any other factors that complicated the optimization process?
Another crucial factor was ensuring the drug could actually reach its intended target. Remember, we're not just dealing with a molecule in a test tube here.
Right.
We're talking about a complex biological system, the human body, and getting a drug to the right place, in the right concentration, and at the right time is no easy feat.
So it's not just about designing a molecule that can bind to the virus. It's about understanding how it will behave within the intricate environment of the human body.
Precisely. And to make things even more challenging, researchers also had to think about potential drug resistance. Viruses are notorious for their ability to mutate and evolve, and there was a real concern that the virus could develop resistance to any new antiviral drugs.
So it's like playing a game of chess against an opponent who can change the rules at any moment.
That's a great way to put it. Researchers had to anticipate how the virus might evolve and try to design drugs that would be effective even against potential mutations.
So they had to be one step ahead of the virus, constantly adapting their strategies.
Exactly. It was a monumental task, and it involved a deep understanding of virology, medicinal chemistry, and even evolutionary biology.
This is all starting to paint a picture of just how complex and multi-layered this process is.
It is, and I think it's a testament to human ingenuity that researchers were able to develop effective antiviral treatments for COVID-19 in such a short amount of time.
It's a reminder that even in the face of seemingly insurmountable challenges, science can prevail.
Absolutely, and it all starts with those early stages of hit-to-lead and lead optimization. Those initial steps are crucial for laying the foundation for a successful drug discovery journey.
All right, so we've explored this intricate dance of optimizing a molecule's potency, safety, and drug-like properties. We've talked about the challenges of navigating the human body and outsmarting a constantly evolving virus. But before we move on, I'd love to dig a bit deeper into this idea of using computer simulations to predict which molecules might be most promising.
Ah, yes, the world of predictive modeling. It's a fascinating field that's rapidly transforming drug discovery.
So how exactly does this virtual exploration work? What kind of tools are researchers using to predict the behavior of these molecules?
They're using a variety of computational models, each with its own strengths and limitations. Some models are incredibly detailed, focusing on simulating the interactions between a drug and its target at the atomic level.
So it's like having a high-powered microscope that can zoom in on the tiniest details of that interaction.
Exactly. They can visualize how the drug molecule fits into its binding site, how it interacts with specific amino acids, and even how those interactions might change the shape or function of the target.
That level of detail is incredible.
Yeah.
But I imagine there are also models that take a more zoomed-out perspective, looking at how the drug behaves within the larger context of the body.
Right. There are models that focus on predicting a drug's pharmacokinetic properties, things like its absorption, distribution, metabolism, and excretion. They're essentially trying to simulate how the drug will travel through the body, where it will accumulate, and how long it will remain active.
So it's like having a virtual map of the drug's journey through the body.
That's a great way to put it. And these models are becoming increasingly sophisticated, taking into account factors like blood flow, organ function, and even individual genetic variations. They can help researchers identify potential roadblocks early on, like a drug that's poorly absorbed or rapidly metabolized.
So it's a way to identify potential problems before they derail the entire drug development process.
Exactly. And then there are models specifically designed to assess a drug's potential toxicity. They can flag potential red flags like interactions with other drugs or off-target effects that could lead to adverse reactions.
So it's like having a virtual safety net that can catch potential problems before they harm a patient.
Precisely. And these safety assessments are crucial for ensuring that only the most promising and safest candidates move forward in the development process.
This all sounds incredibly promising, but I imagine these computational models aren't perfect. There must be limitations to what they can predict.
You're right. No model is perfect. They're based on our current understanding of biology and chemistry, which is constantly evolving, and they rely on experimental data, which can be incomplete or even contradictory at times.
So it's important to remember that these models are tools, not crystal balls. They can provide valuable insights, but they can't predict the future with 100% certainty.
Exactly, and that's why it's so important to use these models in conjunction with empirical data, the results of actual experiments. The two approaches complement each other, providing a more complete and reliable picture.
So it's a constant back and forth between the virtual world of simulations and the real world of laboratory testing.
Precisely. And as our understanding of biology and chemistry advances, and as computational power continues to increase, these models will become even more accurate and powerful.
This all leads to another question that's been on my mind. We've talked about these computer simulations as a way to predict the behavior of existing molecules. But could they also be used to design entirely new drugs from scratch?
That's a great question, and the answer is a resounding yes. In fact, that's one of the most exciting frontiers in drug discovery, the use of artificial intelligence, AI, to design completely novel drug candidates.
AI designing drugs, it sounds like something out of a science fiction movie.
It does, doesn't it? But it's becoming a reality. AI algorithms can analyze vast data sets of chemical structures and biological activity, identify patterns, and even generate new molecules that have the desired properties.
So it's like having a virtual chemist who can sift through billions of possibilities and come up with new chemical structures that we humans might never have thought of.
Exactly. And these AI-designed molecules can then be synthesized and tested in the lab just like any other drug candidate.
This is blowing my mind.
Mhm.
So AI is not just speeding up the drug discovery process, it's actually expanding the possibilities of what's possible.
Precisely. And this is just the beginning. As AI technology continues to advance, I think we'll see even more groundbreaking applications in drug discovery.
This is all incredibly exciting. It feels like we're on the cusp of a new era in drug development. But before we get carried away with the possibilities of AI, I'd like to circle back to a topic we touched on earlier, the challenge of drug resistance.
Ah, yes, the constant battle between pathogens and the drugs we develop to fight them.
It's a reminder that even as we develop new and innovative therapies, the pathogens are constantly evolving, always one step ahead.
It's a constant arms race, and it's one that we can't afford to lose.
So how can we stay ahead of these evolving pathogens? Is there a role for AI in combating drug resistance?
Absolutely. In fact, AI is proving to be a valuable tool in this fight. AI algorithms can analyze the genetic sequences of viruses and bacteria, identify mutations that are associated with drug resistance, and even predict how these pathogens might evolve in the future.
So it's like having a virtual evolutionary biologist who can predict the next move of the enemy.
That's a great analogy. And this information can then be used to design drugs that are less likely to be affected by resistance. For example, AI can be used to identify multiple targets within a pathogen, increasing the chances of developing a drug that will remain effective even if the pathogen mutates.
So it's like a multi-pronged attack, hitting the pathogen from multiple angles, making it harder for it to develop resistance.
Exactly. And this approach is becoming increasingly important as drug resistance becomes a growing threat to global health.
This has been a truly fascinating discussion. We've covered so much ground, from the intricate dance of optimizing a molecule to the mind-boggling possibilities of AI-driven drug design. It's clear that drug discovery is a complex and ever-evolving field.
It is, but it's also incredibly exciting. We're constantly learning new things, developing new tools, and pushing the boundaries of what's possible.
And it's all driven by a common goal to improve human health and alleviate suffering.
Absolutely. That's the ultimate motivation for all the hard work and dedication that goes into drug discovery.
I think this is a great place to pause for now. We've explored the fundamentals of hit-to-lead and lead optimization, delved into the challenges of drug resistance, and even glimpsed the future of AI-driven drug design.
Sounds good. Welcome back to our deep dive into drug discovery.
Yeah, we've covered a lot of ground already.
We have, exploring how to turn a promising hit into a potent lead. Even took a peek into AI-driven drug design.
It's really amazing stuff. It is. But now, let's get out of the theoretical and into the real world.
Sounds good.
Real-world examples. You mentioned SARS-CoV-2 antivirals before.
Yes.
Can you walk me through a success story or even a case where optimization hit a snag?
Of course. The race to develop COVID-19 treatments, it was a real roller coaster.
Yeah.
Full of both triumphs and setbacks. One example that shows how complex lead optimization can be is molnupiravir.
Molnupiravir. I think I remember hearing about that.
Yeah.
Wasn't that one of the first oral antivirals?
Yes, one of the first oral antiviral pills for COVID-19.
I see.
It was initially seen as a potential game changer.
Wow.
A small molecule that stops the virus from replicating.
Okay.
And early studies, they showed some promising results.
So it had real potential.
It did, but there were some hurdles.
Of course.
One of the biggest challenges was its safety profile.
Oh.
Early studies suggested it might be mutagenic, meaning it could potentially cause changes to DNA.
That's a huge concern, especially for widespread use.
Absolutely. Researchers had to carefully weigh the benefits against the risks.
Right.
They conducted tons of studies to assess its mutagenic potential and to figure out the right dosage and treatment length to minimize risk.
So a real balancing act, trying to use the drug's power while making sure it's safe.
Exactly. And this shows just how crucial lead optimization is. It's not just about finding a molecule that works, it's about refining it to make it as safe and effective as possible.
I see. So in this case, were they able to mitigate those safety concerns?
To an extent, yes. Further studies showed the mutagenic risk was lower than initially thought, especially with a short treatment duration and the recommended dosage. Regulatory agencies ended up authorizing it for emergency use, but with precautions.
So it became a useful tool, especially early in the pandemic when we had fewer options.
Yes. But it also reminds us that drug development is rarely straightforward. There are always trade-offs.
Yeah.
And even promising drugs can face unexpected challenges during optimization.
So even when something seems like a sure thing, it's not over till it's over.
Right. It shows how important rigorous testing and careful evaluation are throughout the entire process.
That makes sense. But not all drugs are successful, right?
Right. Not every promising lead makes it to the finish line.
What are some reasons why drug candidates might fail during optimization?
Well, one common reason, as we saw with molnupiravir, is toxicity.
Right.
Sometimes a drug that works well against its target might also have bad side effects. It's that balance between effectiveness and safety again.
It's like trying to tame a wild beast. You want its power, but you don't want it to turn on you.
I like that. That's a good analogy. Another reason is poor pharmacokinetic properties. Remember, a drug can't just be potent in the lab. It needs to work inside the human body, be absorbed, reach the right tissues, be metabolized properly, and then be eliminated without causing harm.
So even if it hits the target, it might get lost, break down too quickly, or build up in the body.
Exactly. And these pharmacokinetic hurdles can be super hard to overcome. Sometimes, no matter how much you tweak the molecule, it just won't behave.
So we're up against the complexity of the human body.
We are.
It's hard to predict how a drug will act with all its systems.
Precisely. And then there's drug resistance, which we talked about earlier.
Right.
Some diseases, especially infectious diseases, those pathogens, they can evolve. They can outsmart even the most potent drugs.
Like a constant game of cat and mouse. They adapt and find ways to survive.
It is. And that's where understanding evolutionary biology is key. Researchers need to think ahead, predict how a pathogen might mutate and become resistant. They need to design drugs that can stay one step ahead.
So it's not just about hitting a target, it's about predicting the target's next move.
Yeah, like playing chess against a grandmaster.
It sounds like a real detective story, gathering clues, analyzing evidence.
It is, and it shows why collaboration is so important in drug discovery. It's not a one-person job. It takes a whole team, each with their own expertise.
Chemists making the molecules.
Yes.
Biologists studying how they interact.
Pharmacologists seeing how they behave in the body.
And even computer modelers.
Exactly. All working together to solve this complex puzzle.
This brings up another question. We've talked about potency, safety, drug-like properties. How do researchers actually decide which is most important? Is there a set order, or does it depend on the drug and the disease?
That's a great question, and there's no simple answer. It really depends on the situation.
Okay.
For example, a life-threatening disease with no current treatments. You might be willing to accept higher risks for a more potent drug.
So the potential benefit outweighs the risks.
Right. But for a chronic condition where there are already treatments, safety might come first. You wouldn't want to introduce a new drug with serious side effects if there are already safer options.
That makes sense.
It's nuanced. And it involves ethics, too.
Oh, of course.
Researchers have to think about the benefits for patients versus the risks, and their decisions must align with ethical guidelines.
Drug development isn't just science. It's about making responsible choices for patients.
It absolutely is. The human element is always there, at the heart of it all.
I'd like to understand more about how they actually optimize a lead molecule. What do they use to make those improvements?
Well, it uses both experiments and computer modeling. One basic tool is structure-activity relationship studies, SAR, which we mentioned before.
Right, SAR.
It's all about how changes to a molecule structure affect its activity.
Like those molecular architects.
Exactly. Chemists can make small modifications to the molecule, adding or removing atoms, changing the arrangement of functional groups, even adding entirely new parts.
They're basically testing different versions, trying to find the best one.
Right. And they use a lot of different techniques for these SAR studies. One approach is combinatorial chemistry,
Okay.
where you make a huge library of molecules, all with slight variations. Then you screen them all for the desired activity.
Testing hundreds or even thousands of molecules at once.
Exactly. And that can be efficient, finding promising candidates quickly, but it can also be pricey and take a lot of time.
Speed versus cost.
Right. Another approach is structure-based drug design. This uses the 3D structure of the target molecule to design new drugs.
So they have a blueprint of the enemy, and they design a weapon to target its weak spots.
Exactly. Researchers can see how the molecule interacts with its target at the atomic level. Then they make changes to improve things like binding, affinity, and selectivity.
So it's much more precise compared to combinatorial chemistry.
It is, and it can be very powerful, especially when we know the target structure and how it works.
It sounds like lead optimization is a back and forth between experiments and computer models.
It is, and it shows human ingenuity and perseverance. Despite the challenges, we've come a long way in developing treatments, and I'm excited to see what the future holds.
Me too. It's a field of possibilities, driven by wanting to improve human health.
That's what makes it so compelling.
This has been really enlightening, this journey through lead optimization. We've seen the challenges, the wins, the science, and the ingenuity. And we've seen how, even though it's complex and demanding, it's all about helping people, improving lives.
It is.
I think this is a good place to pause.
Okay.
In the last part of our deep dive, we'll talk about some broader things in drug discovery. The ethical, societal, and economic factors at play.
I look forward to it.
Me too. I'm sure our listener does as well. Welcome back to the deep dive. It's been quite a journey so far, right?
It has, from those early hits to the whole process of lead optimization.
We've uncovered so much. The science, the challenges, the victories, everything that goes into developing new medicines.
It's been fascinating. I think we both have a much better grasp now of how complex it is to turn a promising molecule into a safe and effective therapy.
We've talked about a lot of the technical stuff, the molecular changes, the simulations. But as we wrap up, I'd like to step back a bit. Look at the bigger picture. Because it's not just about molecules in a lab, right?
Absolutely not. Drug discovery is really about people. It's about easing their suffering, improving their lives, helping them live longer. And that means thinking about how our decisions affect people through the whole process.
I like that. So shifting from the lab to the real world, what are some of the big things that shape drug discovery?
Well, money plays a huge role. Drug development is incredibly expensive and risky.
Yeah, I can imagine.
It often takes years, sometimes decades, to get a new drug out there.
Wow.
So it's not just about scientific discoveries, it's about finances too.
Oh.
The research, the clinical trials, getting approval, marketing, it can cost billions.
Billions?
And there's no guarantee it'll work out.
Oh, wow.
Many drugs fail along the way, and companies lose huge amounts of money.
So it's a big gamble.
It is. So companies have to balance the need for innovation with the financial reality.
I bet that's tough. Trying to make life-saving treatments accessible and keep the company afloat.
It is. It's a tough situation, and there's no easy solution. But we need to talk about it. We have to find ways to encourage innovation while also making sure essential medicines are affordable for everyone.
It's a reminder that drug discovery isn't isolated.
It's not.
It's connected to economics, markets, even politics.
You're exactly right. And as we think about these bigger issues, we can't forget about regulatory agencies. They're crucial. They make sure new drugs are safe and effective before anyone can use them.
So they're like gatekeepers, protecting public health, making sure only the good drugs get through.
Exactly. They set high standards for trials, study the results, and decide if a drug's benefits are worth the risks.
It's a big responsibility. I'm glad they do it. But that process must slow things down
Right.
and make it more expensive.
It can, and that can be frustrating for patients waiting for new treatments. But it's a balance we need. We have to make sure new drugs are thoroughly checked before they're available.
It's not just about being fast. It's about being careful and putting patient safety first.
Absolutely. And in all this, we can't forget the patients themselves.
Of course not.
They're the ones who matter most. They're the ones who benefit or are harmed by the drugs. We need to hear what they have to say.
Patients are at the heart of it all, so it's important to involve them.
Right. They have valuable insights about their experiences, their needs, what they think about the risks and benefits.
It's a two-way street. Researchers need to listen to patients, and patients need to speak up and be part of the decisions.
Exactly. And as we finish up, I think it's amazing how far we've come in drug discovery.
It is?
We've created life-saving treatments for so many diseases, help people live longer, healthier lives.
It shows what we can do with ingenuity, persistence, and collaboration.
It does. But we can't stop here.
Right. There's always more to do.
There are still diseases without effective treatments, and new challenges are always appearing.
So the search continues. We'll keep looking for new and better medicines to ease suffering and improve health.
And I think as long as we keep asking questions, keep learning, and keep patients at the forefront, we'll keep making progress.
That's a great message to end on. Thank you so much for sharing your knowledge with us. It's been an incredible journey.
It's been my pleasure. And to our listener, thanks for joining us on this deep dive. I hope you've learned a lot about this important field. Keep exploring, keep asking questions, and keep believing in the power of science.
Until next time, on The Deep Dive.